Debates around gain-of-function research, or research that genetically alters an organism to give it new functions, became more public and much more charged during the COVID-19 pandemic, when public figures began to speculate that the virus responsible was created through gain-of-function experiments in Wuhan, China. But many scientists maintain that this type of research helps us to understand and respond to viral threats like the H5N1 virus or bird flu, which is continuing to spread through the United States agricultural industry and across the globe. So just how important is gain-of-function research, and what oversight mechanisms are in place, whether within the United States or internationally, to reduce the hazards that make these experiments at least potentially risky? On the podcast to discuss all this today are Kirstin Matthews and Alicia Johnson, respectively a fellow and postdoctoral associate at the Baker Institute's Science and Technology Policy Program, and Yousif Shamoo, a professor of biosciences at Rice University, as well as a fellow of the American Association for the Advancement of Science. Kirstin's and Alicia's work focuses on the ethical, legal, and social implications of synthetic biology, while Yousif's lab at Rice is engaged in the response to the rise of antibiotic-resistant bacteria. I'm David Satterfield, director of Rice University's Baker Institute for Public Policy in Houston, Texas, and this is Baker Briefing. Thank you all for joining me today, and Yousif, let's start with the basic science here. How do you define gain-of-function research to those of us outside the scientific community? What's it meant to do?
Well, first off, thank you for having me on the podcast. Gain-of-function research is actually an incredibly wide field. Recently, we've heard a lot about gain-of-function in the context of viruses and pandemics and things like that. But gain-of-function is actually already in place in many technologies in our life already. It's the ability to give either a bacteria or an animal or a plant a function they didn't have before. That is what a gain-of-function experiment is. It's just giving an animal or a plant or a virus or a bacteria something it didn't have before. And that can be very innocuous, or it could be potentially more risky. And so the risk-reward ratio is the critical thing in determining whether you should do a gain-of-function experiment.
And just to clarify a bit further, genetic modification of plants, is that a gain-of-function, or is that a different issue?
That's a gain-of-function. If you gave the plant the ability to not be killed by a herbicide, like the Monsanto product, that is a gain-of-function. That plant was not resistant to that herbicide before it was genetically engineered.
What are some of the major hopes scientists have for gain-of-function research going forward? What kinds of applications beyond the one we just noted does this research have?
Sure. I'll take it from things that I think are just amusing to things that we would think all agree are dangerous in terms of gain-of-function. The first example is there's a company that has made glowing petunias called firefly petunias. What's unique about them is they're petunia plants, and when you turn off the lights, they glow. So it's just amusing. It's not dangerous. But the way they did it is they engineered it by taking part of the genetic material from a mushroom, and they put it into a petunia. Now, this mushroom had the ability to sort of glow in the dark. I don't know why. But when you put it in the petunia, now the petunia glows in the dark. So that's just, I would say, a novelty. Another way that it's been used is in the production of insulin. So insulin is something, of course, we all know is used in the treatment of diabetes and is very important for medical purposes. So the way insulin is made now is essentially, again, taking a small amount of genetic material from the human genome and putting it into a bacteria or a yeast. You give the bacteria or the yeast the ability to make insulin. The risk there is basically zero, and the reward is fantastic, right? You make insulin cheaply and make it very available. So that's actually gain of function because the bacteria normally doesn't make insulin, right? Now, the last example I'll give you is one I think we would all agree is the worst type of gain of function we can probably imagine, which is that in the late 1980s, the Soviet Union actually modified the plague bacteria, Yersinia pestis, which killed millions of people in the Middle Ages, to make it more lethal and harder to treat clinically. That's something that is the worst application of science I can think of, which is to make something much more dangerous than it was previously. So that's the spectrum. You go from things which are completely innocuous to things that are very useful to things that can be very dangerous.
Yousif, on that last point, let's go in the opposite direction. If one engineers an organic virus, bacteria, to make it less able to defend itself against treatments, is that gain of function?
That's loss of function, and it's actually a very important medical and scientific approach to problems. For instance, if you make a drug-resistant bacteria now sensitive to the drug, you've done a good thing. Now the MD can treat you with an antibiotic that they couldn't treat you with before. They're related. They're just mirror images, gain of function, loss of function.
Kirstin, we saw a lot of speculation. It's not a past tense. We continue to see a great deal of speculation, both during and following the COVID-19 pandemic, about where and how that virus originated, with many insisting, including quite recently, that it emerged from a lab leak after being created through gain-of-function experiments at the virology lab in Wuhan, China. 5 years after the pandemic, what do we actually know or don't know about how this virus originated from where?
So this is kind of a complicated problem because part of it is kind of unknown and almost unknowable. And there's two reasons. Most of the scientific data we've looked at points towards this actually being a result, the virus, of animal transmission to a human. And it comes from the markets within Wuhan is what we estimate. But there is still a small level of uncertainty. And unfortunately, this is due a lot to the lack of transparencies within the labs in Wuhan, right? We weren't able to go into those labs. It took months to go into them. Not all the information, the people were accessible when the scientific community went there. And so that left some of it unaddressed is the best estimate that it came from an animal. But we will never be able to cleanly understand where it came from because of that lack of transparency. I will say that it didn't come from the US because we have a lot more transparency for issues like this.
And how has this uncertainty had an impact on how scientists approach or don't approach gain-of-function research today?
So I wanted to talk a little bit about right now in the US, how we do gain-of-function research, how we do, especially the ones that Dr. Shamoo was mentioning, the ones that we're questioning, like virology, potential pathogenics. We actually have a lot of regulation oversight of that research. There's lots of paperwork you have to do to be able to do this. There's lots of training that's involved. There's special equipment and special facilities to be able to do this work. Our scientists, I think, are quite conscious about what they're doing. We have government oversight of this. And then we have transparency. So I think a good example would be one of the lab leaks was anthrax at one point. And we were able to actually trace the anthrax in what lab we felt like it came from because of the transparency of looking at what the product was that went in the mail, but also within the labs and be able to test and do comparisons to those. That is uniquely, I think, more US oriented, where we weren't able to do that in China at the time. It was also at a time where everything was in flux. So it took a couple of months before we could get the ability to trace things like that. But it was still a lot more transparent in the US.
I just want to chime in a little bit with what Kirstin said. So we're not ever going to know what happened in Wuhan because of the lack of transparency of the Chinese government and the Chinese investigation. So unfortunately, I think we're never really going to know the answer to this question. And that's not what would happen in the United States if something like that were suspected. In the US, you would actually be interviewing the scientists, looking at their records, and it would be open and known to everybody, essentially, because of the NIH oversight. So I think we're probably never going to get the answer, but it could have been as simple as like one of the people collecting these wild viruses may have infected themselves when they gave it to the Wuhan Institute, right? It doesn't have to be something nefarious. There's no evidence that the COVID-19 was engineered to be a plague virus, right? There's no evidence of that, but that doesn't mean somebody in the field or maybe in the lab didn't accidentally give themselves the COVID.
And would we know whether a virus such as COVID-19 was in fact specifically engineered?
So most intelligence agencies have deemed that it was not engineered. There would be scars. There's indications that when we manipulate DNA, because we're not nature, we tend to leave man-made, fingerprints on the DNA that we've manipulated. Now, could somebody nefariously try to erase those marks? Yeah, but COVID-19 is not a good weapon. There are much better weapons from the Cold War. COVID's a terrible biological weapon. It doesn't even make sense to engineer it. So I think the probability that it was engineered is basically zero. But that doesn't mean that somebody didn't accidentally get contaminated at the Wuhan market, or maybe somebody who worked at that lab potentially, because the oversight in China is not as strong as the United States.
On that theme, Alicia, how do scientists assess benefits of gain-of-function versus risks of gain-of-function research? And what exactly are those risks other than accidents?
So to answer your second question first, what are the risks of gain of function? So there is the possibility for accidental release, but that possibility is very low due to the many oversight mechanisms that we do have, as my colleagues were mentioning. And it's worth noting, too, that most gain of function experiments that are occurring are not inherently risky and they don't involve pandemic pathogens. The bigger concern that we should be having at this time is with the rapid changes and the cuts that are happening in our government could lower the strength of the oversight that we do have. And as far as how do scientists assess, it's important to note that scientists do not do this alone. There are many players involved from every step from when you write the grant to when you do the first experiment. So this can include your NIH program officer. If you do have a grant that involves pandemic pathogens, it goes through an enhanced strict review process. There's human subjects oversight at the institutional level, at the university level. You have to submit every protocol that you're going to do in your lab for it to be approved. And so there are a variety of mechanisms that help scientists assess the risk and the benefits of their experiments.
And for both you, Yousif, and you, Alicia, the broad issue, where are we in terms of federal policy right now? And I realize this question touches on something that's very much dynamic and in flux, but has there been a specific discussion of synthetic biology or gain of function research since this administration took office?
So there are a variety of discussions that are still ongoing from the previous administration into this administration. So a lot of policies actually got updated in 2024, like dual use research of concern, DNA synthesis frameworks, NIH guidelines, so on. And
what is dual use research?
Research that could be used for benefit, but it could also be used for potential nefarious purposes.
And I agree with what Alicia said. Under the Biden administration, there were some new protocols put into place and some new policies implemented. They're actually coming into effect in May, as far as I know, still. Under this administration, which actually put a lot more scrutiny into what we would call gain of function research, where the risk reward, where the risk is actually significant potentially. So there will be an enhanced scrutiny coming from the previous administration, which seems to be being continued into this administration. That's an important thing to bring up, that actually there have been some changes recently to give more oversight even.
And moving to bottom lines here, were there to be strictures imposed beyond those already present or contemplated to come into effect, or a ban on gain of function research, what impact would that have on science and the ability to respond to viruses and the threat viruses pose? How fundamental is gain of function research itself to virology and to the treatment of disease?
If you just banned gain of function, even if you banned gain of function had some risk associated with it, you would essentially cripple the biomedical industry going forward. So as we've seen recently, there are actually genetic engineering vectors to actually cure sickle cell anemia, which are all based on genetic engineering of cells in the human body of the patient. So I think most people would think that's a good thing. We've seen applications of mRNA vaccine being used to potentially cure types of cancers. And so it goes beyond looking at biosurveillance of viruses. Actually, these genetic techniques are being used in lots of medical applications. So if you were to ban it outright, all you'd be doing is handing it to other countries to develop these important biomedical breakthroughs.
I was actually just going to add, I think what we need is this oversight to make everything more comfortable. But right now, the one policy the administration has done, which is reduce the number of individuals at a lot of these agencies that would provide this oversight. And I think that's kind of what we worry about the most, is the oversight allows us to do the research in a way. So a principal investigator, so that's the head researcher in a lab, really wants to do what's right. They don't want to accidentally release it into the population. They don't want to do something that's unethical. And so having a really oversight and having these conversations, and we need someone at the government to be able to have those conversations to ensure we're moving forward in an ethical way. Everyone wants that. The guardrails are in place to help the public understand what we can and can't do and feel comfortable with that, but also make the scientists feel really comfortable with what they can and can't do as well.
And I guess this is a moment to try to drill down on an issue which is much broader than synthetic biology or this field. The cuts you referred to, is there any known specific justification for cuts in a given area, synthetic biology protections, or is it a broad agency or function-wide set of cuts applied that have the impact? In other words, you're reducing staffing of 10,000 people by 60% as opposed to targeting specific staffing in specific functional or operational areas.
So far, what we're seeing is just a blanket cut, right? We're just seeing someone just taking and saying, you have a group of 10 people, half of those have to leave, or they actually decide those people we think have to leave. It's interesting enough also to see that people have left and then come back because they realized that in that 15-minute interview, they didn't fully understand what the person's job was. And we do want to have our nuclear warheads safe, so they had to come back in order to protect the nuclear stockpile. And that applies, I think, to a lot of areas in government. And then in the science, I think sometimes we're not the best at explaining our jobs and what we do. So that's a little bit of our problems of not being able to communicate effectively. There are probably places of inefficiency in the government. I'm not going to pretend like we're really efficient. So I can see a place for going through systematically and having those conversations. Okay, maybe you don't need 10 people. I think you can work with 8. Then this group can work with 2 conversations. But right now, we're just getting broad cuts. And those end up making a small number of people do more work. And they're able to do it less effective because they have an increased workload.
I absolutely agree. Like as Kirstin said, we all agree that there's places where the government could be made more efficient. And I think everybody's for that, right? But I think just sort of randomly laying off people is not the way to do it. I think that there could be some thoughtful analysis about looking at who's essential and who's not essential. To take away, for instance, the regulators that are looking at biosafety and biosecurity doesn't seem like a really great idea given what we're talking about today. You want to have oversight. The scientists themselves want the oversight. When I am doing an experiment in my BSL-2, our biosafety level 2 laboratory, right, which works with pathogens, I want the university to look at my work. I want to have the NIH look at my work. I want the DOD to look at my work if I'm doing DOD work, to tell me that this is the right way to do it, because I don't know everything. Nobody knows everything. So it helps me to have experts, all co-experts, tell me this is a good experiment, it's worth doing, and we're going to give you a green light to do it.
Alicia, what should we do to mitigate the balance between risk and practical application or vital application of gain-of-function research? How do you strike the balance?
Well, it's a delicate balance to strike, but going back to this concept of risk and with what we have right now, the biggest risk that we do have right now are the broad cuts that have been made. Because what makes oversight mechanisms effective is that you need enough people that have the technical knowledge and are able to enforce them. And going back to how we strike that balance between risk and benefits, again, scientists, there's a whole team of people that are helping scientists do this.
Yeah, and I actually, to talk a little bit more about synthetic biology, I was just at a conference in Asilomar, California, that was put together by a Rice historian, Luis Campos, which featured next generation leadership, prominent expert scientists, social scientists, people from the global south, a broad group of people to talk about synthetic biology in this context. And one of the things we talked about was if you're doing synthetic biology to modify a plant to make it a plant that can fix nitrogen so it can fertilize itself, which would be immensely awesome because we use 2% of the world's energy is used to make fertilizer. That would actually help us for climate change, right? We could actually produce less CO2 if the plants could just fertilize themselves by taking nitrogen from the air and putting it into their roots. And this is a synthetic biology operation. But in that context, we're going to take a plant out of the lab and we're going to put it out in a field. So you need to have, regulations and guidance and oversight of this idea that you're going to put something in a test field. And that's a little bit different because normally biosafety is about keeping everything in my laboratory. But in this case, it's going to go out into the field. And that requires different types of oversight and different kinds of thinking. That was a big part of this conference. I talked about the idea of environmental safety levels. How do we talk to people in the neighborhood? How do we talk to the stakeholders? So it's more than a scientific discussion. It's a very broad question. But synthetic biology is going to be in the world. That's a big difference between what's happening in my laboratory where I'm studying a pathogen in my lab, and it's definitely not leaving my laboratory, versus one that's going to definitely be leaving my laboratory.
And in that world you just described, And staying on the theme of agriculture, two common concerns are raised. First is that this commoditizes products. Farmers are now required to buy a specific product, which previously they could produce themselves through natural seeding. Now they must reacquire for each crop the proprietary item to plant. So there's an economic impact. There's a benefit, but an impact. The second is, and you all may want to speak to this one, you have started a chain of, quote, mutation in a natural thing. Who knows where it ends up? Who knows where the glow-in-the-dark petunia takes us? Now, that's basically an economic argument, not a scientific one. But on the second point, what is the risk of introducing a change, gain of function or loss of function, either one, which then in and of itself becomes the initiator of further potentially harmful changes? Does that risk exist?
I think the risk is very minimal in the context of the products we've described, because nature is the greatest experimentalist out there. Right? Mutations are happening all the time in the natural world. Genetic materials being moved from one organism to another willy-nilly in this world by nature. That happens all the time. But that doesn't mean there's no risk either by nature, because when nature experiments, sometimes we get a new flu. That's not good. But you could also imagine in synthetic biology, we put something out in the field and maybe it has an unintended consequence. Maybe it ends up affecting the insect life in the region or something like that. This is why you need to do a lot of thought. But in the end, synthetic biology is going to be out in the fields more than it's going to be in the lab. And I think that's a really interesting challenge that you've put out there. The commoditization of these resources is not my area, so I'm going to leave that to the experts.
Well, I'm from rural Kansas, so having an uncle who specifically talks about this on a regular basis, hi, Uncle Gary, it is going to be a really big challenge because those are individuals that work almost from paycheck to paycheck. And this has actually been really hard to have one. And I know that it's not the scientists that are really, truly interested, at least, you know, the Dr. Shamoos of the world, in commodifying it at a level that it can't be sustainable. I think our scientists, compared to when we started with GMOs, are now more thoughtful on also developing these technologies to be sustainable. And sustainable also means that you don't have to purchase them on a regular basis, that they would perpetuate on their own and help the environment on its own. So the goal isn't to keep creating the bacteria, because that also creates pollution and resources. It is the reason why we would even be interested in a bacteria that did these things with the nitrogen, is so that it would happen naturally and they would stay potentially in the soil for a lot longer.
Final question on substance of research direction. Is there any gain of function research on human cells? That is, to produce, not for a plant, not for a bacteria or a virus, but in the human body, a synthetically introduced new function?
So this is a very tricky and important ethical question. Very, very important. Maybe one of the most important ones to come up in science in its history. Now, I think most of us are okay with getting a cancer therapy or cancer treatment that cures, let's say, our pancreatic cancer. That's something I've decided to take, a medicine I've taken, whether it's a vaccine of sorts or whether it's a, just an anti-cancer drug. I take it, I cure my cancer, hopefully, and I go on. What's really different is you take my germ cells, that is to say my sperm or my partner's egg, and you modify that, because then the children are modified in perpetuity. That would be a type of gain of function that I think is quite the slippery slope that we're going to have to deal with as a community and as a world. It's already out there. We saw one case illegally in China where 2 children were genetically modified, actually 3 children now were genetically modified without proper oversight or regulation. And so this is going to be an important discussion. I personally, I'm not excited by the idea of having designer children, because I don't, again, talk about commodification. Who's going to be able to afford this, even if you could do it properly? So I think it would become a very, very bad outcome if we didn't have oversight and serious thought about whether we should ever be doing this. But it's hard to put a genie back in a bottle, I'm told.
And Kirstin, on that note, science itself, the existence of verifiable fact, all of this is under challenge now. Not for the first time in human history, but definitely now. How does science, in a structured sense, an organized sense, and those involved in science, that broad community, go about explaining better what they do, how they do it, what the risk-value factors are, and what is done to try to restrict the risk side and enhance the value side of that equation?
So this is a really challenging problem. For a long time, scientists were told by people that we weren't supposed to talk about risk or we weren't uncertainty. But what we found with the pandemic is that it was all uncertainty, and why not actually communicating it effectively when policies changed with the science that people didn't understand why. But also there's a lot of mistrust, I think, of the government, but also science that is really framed around misinformation. So some it's deliberately, I'm just saying a falsehood. Sometimes it's taking something completely out of context and quoting it as a fact, but not knowing what that actually means, or just misunderstanding the data. They read something online and they say, this is what it really means. We try to correct the information, but I think we need to go a step further, which is actually really have a deeper conversation about it and change the conversation more. So if you're saying, I don't like gain of function research, it's not that you're combating the data they're showing because it's not true. It's more of like, this is actually a really bedrock part of our bioeconomy. We see this as really a future for not just medicine, but agriculture. And by limiting it in the US, we're just going to hand over all of our technology to other countries and then later on be paying their corporations as we use it in the US. We would like it actually to be in the United States because the biggest funder of biomedical research in the world is the National Institutes of Health. Like by far, it beats every other country. It beats the nonprofits. What we give out each year really promotes a really high level, and they say this is how you should do it ethically. And that actually sets a really high standard because everyone actually kind of raises themselves up to that level of what we say in the US to do. And I think it's really important as us as a player to still be participating in this.
This will undoubtedly be a subject or a set of subjects for further discussion as administration policies crystallize, as the global community responds to developments here in the United States, and as both the presence of disease and risk factors and the ability or inability to deal with them emerge. None of this, as all of you have made clear, is static. It's dynamic, and it is not something that could be controlled by an executive order or indeed by any one particular clinical or scientific step. It's a constant process of awareness, response, awareness, response. But thank you all very much for your contributions. Baker Briefing is brought to you by the Baker Institute for Public Policy, which provides meaningful, nonpartisan policy analysis on the most critical challenges facing the United States, Texas, and the world. This episode was produced by Karina Zamel with support from Maggie Siganova. AV production was led by Kevin Young. Be sure to visit us at bakerinstitute.org to learn more. Thank you for listening.